Let's zoom in a bit on muscle structure now. So you've got a whole muscle here. The muscle is surrounded by a connective tissue called fascia, which is kind of translucent. That's this material that you see back here. That fascia is actually continuous with the tendon. That tendon is very tough connective tissue that connects the muscle to a bone. And as the tendon comes and reaches the bone, it actually spreads out over the bone and becomes this thin layer of connective tissue that covers the entire bone. It's called periosteum. Now, if we look at the muscle itself, we have these fasciculi, which are bundles of individual muscle cells or muscle fibers. Here is a single muscle cell, or muscle fiber. It means the same thing. Now we're going to zoom in on that. So here we are down at the bottom picture. This is the single muscle cell, and you can see there's a nucleus here. It's multinucleated. There's another one here, and of course there would be more and more down the whole length of this thing. Inside of that individual muscle cell, we have myofibrils, which are bundles of myofilaments. The myofilaments are the actin and myosin proteins that make the muscles contract. So here are those thick and thin filaments. The thick filament is the myosin. Myosin is a protein that's made out of two polypeptides that kind of wind around each other. Each myosin molecule looks a bit like a golf club. It has a head and a long handle, and they clump together more or less into the shape you see in the middle of this slide to form the thick filament. The thin filament is mostly actin, but it's also got tropomyosin and troponin, a troponin complex on it. And the tropomyosin and troponin complex together are part of the regulatory structure that can regulate when muscles contract and when they don't. This is the sliding filament model for how muscles contract. So we know that we've got these thick and thin filaments. So basically actin are the thin filaments for the most part, and myosin are the thick filaments. And so if you've got those two kinds of proteins in there, how does the muscle get shorter? And one hypothesis might be that somehow that those protein molecules themselves get shorter, and that's what makes the muscle contract. But in reality, none of the proteins actually change length. The way that it works is that the thick filaments essentially slide along the thin filaments. They pull themselves along with the heads on the myosin, and that's what makes the muscle contract. And actually that also explains why muscles get bigger around when they contract, because nothing is going away. All you're doing is making these fibers slide past each other, so they have to go somewhere. The volume stays the same as the muscle gets shorter, so it has to get bigger around. If you were to zoom in on one of those myofibrils right here, you can actually see this with a microscope. There is a line here and here. That's called a Z-line, and the space between those two lines is called a sarcomere. The sarcomere is the contractile unit of a muscle. So basically when the muscle contracts, those Z-lines get closer together. And if you look, well, here's another Z-line. So this is another sarcomere here, and there's another sarcomere here, and another one here, and then they kind of fade out in this drawing. So the Z-lines or the sarcomeres actually get shorter as the muscle contracts. So let's talk about some of the terminology. So if you look at the top picture, that's a muscle that is completely relaxed, and you've got a picture taken with an electron microscope of actual muscle tissue there. Below it is basically a kind of a schematic drawing of what's going on. So the thick filaments are the thick kind of purple lines that are in there. That's the myosin. The thin filaments are the thin golden lines. That's the actin right there. So the sarcomere is the length of a muscle between the Z lines. Here's a Z line, Here's another Z line, so the sarcomere is the part of the muscle between that. And of course, there's another sarcomere on either side of those Z lines. The I bands are the part of the muscle that's kind of looks fairly clear in these diagrams, at least when the muscle's relaxed, and it's right next to the Z line. So that's called the I band, and that's the area where all you have are thin filaments. If you look down here, there's nothing in there except thin filaments. The A bands are where you've got the thick filaments. And so most of that is thick filaments and thin filaments overlapping, but it's this area right here in the schematic drawing, and it's the dark area up here in the actual EM photograph. Those are the A bands. Now remember, when that muscle contracts, none of these molecules, none of these fibers are actually going to get any shorter. They're just going to slide past each other. So there's an important part of the A band, which is right in the middle. It's the part where there are no thin filaments. There are only thick filaments. It's easy to see in the schematic. If you look up here, you can kind of see it, but you kind of have to know to look for it because it's a little bit subtle, but it's a little bit lighter there. And you can see, it probably makes the easiest to look at the schematics, but the top picture has the muscle fully relaxed. The middle picture down here is the muscle as it's starting to contract, and you can see that the H zone here is a lot narrower than the H zone is up here when the muscle's relaxed. And the A band is still exactly the same, right? It hasn't changed width from the top one. However, the I bands are getting narrower as the muscle contracts, and then when the muscle is fully contracted, it's basically just A band. That's pretty much all that you can see. You can still kind of see the Z lines, but it's basically just the A band because everything is overlapping. So let's walk through the steps of a muscle contraction. I will also put up a couple of videos because this is a dynamic process, so I'll kind of walk you through it, but then you definitely need to look at the videos so that you can see things moving and see how it actually happens, and then it'll probably make a lot more sense. So when the muscle's at rest, what that means is that the myosin, the thick filaments, are not binding at all to the thin filaments. That's the muscle completely relaxed. And that happens because there are only certain specific places on the thin filaments where those myosin heads can actually bind. And in this diagram, it's these kind of little purple spots. Let me circle one of them right there. Right there, you see a little purple part on that red sphere. That's a myosin binding site. So as long as those myosin binding sites are covered up, myosin can't bind to actin at all. And it's covered up by the tropomyosin and troponin complex. That covers them up when the muscle is relaxed. Now that's the muscle when it's relaxed. Now we're going to make the muscle contract. So you get an action potential from a motor neuron that reaches the muscle, and there's a little space between the end of that motor neuron and the muscle, and that space is called a synapse, and there's a chemical that gets released into that synapse called a neurotransmitter that basically sends the signal from the neuron to the muscle. So the neurotransmitter is released, gets into the muscle cell. The neurotransmitter causes an action potential in the muscle cell itself. That action potential then makes the sarcoplasmic reticulum release tons of stored calcium ions. Now, the sarcoplasmic reticulum is a storage area that basically penetrates all the areas within the muscle cell. It's a modified endoplasmic reticulum. It's modified just to store those calcium ions. So normally the calcium ions are sequestered away and they're not out in the rest of the cell. When the muscle cell is going to contract, the first thing that happens is those calcium ions are released and they flood the cell. When they flood the cell, the calcium ions bind to troponin, to these troponin complexes right here. The calcium ions are the green balls. And When that happens, it makes the tropomyosin, troponin and tropomyosin complex change shape, and it basically pulls away from those binding sites, the myosin binding sites. So now all the myosin binding sites are exposed. They're not covered up anymore. And the muscle contracts. Now, when the muscle contracts, what's actually happening is that the myosin heads bind to the myosin binding sites on the actin, and then they basically change shape. As they change shape, it's called the power stroke. So here's the next one. Notice how the angle of that head is different. That's called the power stroke. When it's open, like in the first picture, that's in the cocked configuration, and then it does the power stroke. And then what it'll do is release from the binding site, which is what's happening down here, and then open up again. So it's cocked, and then it attaches to the next binding site, and it basically just walks along. So the myosin walks along the actin and pulls it along, and that's why the muscle contracts. Obviously, this requires energy, and the energy comes in the form of ATP, which is the usual way that cells are providing energy to do things. But it's used in, the ATP is used at kind of the timing is a little bit odd. It may not be where you would think it would be. So obviously the power stroke is where the energy is used in this. That is not the moment that the ATP is broken down to ADP and phosphate, however. So if you look at the first picture is the muscles contracting. It says step 2, but it's actually the first picture of the contraction. You notice that that head is already in the cocked position. It's ready to have a power stroke. It's attached to the myosin binding site on the actin. But if you look closely, you see that associated with the head, it says ADP and phosphate. So it's already burned ATP to be in this position. And then when it actually has the power stroke and pulls itself along, once it reaches the closed configuration, it releases ADP and phosphate. Now, here's the thing. If there was no ATP in the cell, this myosin head cannot release from that binding site. And that's actually what causes rigor mortis. When an animal dies, the muscles get really tense because when you die, you're not making any more ATP, and so all these muscles are basically locked in the contracted configuration, and then it'll relax again once things really start breaking down. But as long as the basic structure of the molecules is intact, the muscles are going to be locked in a contracted configuration. So when an ATP molecule binds to that myosin head, the first thing that happens is it then releases from the binding site, and then it changes configuration and goes back into that cocked configuration. And as it does that, that's where the ATP is burned and converted into ADP and phosphate. And now we're ready to attach to the next myosin binding site and continue, pulling along and making the muscle contract. The contraction stops when the calcium disappears. The calcium ions disappear, and the calcium ions disappear when they are pumped back into the sarcoplasmic reticulum. When they're pumped back in, then you don't have calcium around to basically force the binding sites to be uncovered. So the binding sites are covered up, so myosin can't bind to actin, and the contraction stops at that point.